What is microscopy? Microscopy is the scientific technique used to observe and study objects or samples that are too small to be seen with the naked eye. It involves the use of specialized instruments called microscopes, which use various types of lenses to magnify and resolve the details of a sample. There are several types of microscopes, each with different capabilities and applications. Light microscopes use visible light to produce an image, while electron microscopes use a beam of electrons to achieve much higher magnification and resolution. Other types of microscopes include confocal microscopes, fluorescence microscopes, and scanning probe microscopes. Overall, microscopy is a powerful tool that has a wide range of applications in many scientific fields, allowing researchers to study the world at a microscopic level and develop new insights into the natural world. Terminology related to microscopy. 1. Magnification in microscopy. Magnification in microscopy refers to the process of enlarging the size of an object when viewed through a microscope. The magnification power of a microscope is determined by the combination of lenses used, including the objective lens, and the eyepiece or ocular lens. The total magnification of a microscope is calculated by multiplying the magnification of the objective lens by the magnification of the eyepiece. For example, if a microscope has an objective lens with a magnification of 10x and an eyepiece with a magnification of 20x, the total magnification would be 10 times 20 equals 200x. Higher magnification allows for greater detail and resolution of the specimen being viewed, making it an important aspect of microscopy. 2. Resolution. In microscopy refers to the ability of a microscope to distinguish two separate points as distinct and separate entities. It is usually expressed as the minimum distance between 2 points at which they can be seen as separate. The higher the resolution of a microscope, the more detail that can be seen in the specimen being viewed. The resolution of a microscope is influenced by the numerical aperture of the lens, the wavelength of light used, and other factors. The equation commonly used to calculate the resolution of a microscope is known as the Rayleigh criterion, which is resolution equals 0.61x, wavelength of light, numerical aperture, where the numerical aperture, Na, is a measure of the lens's ability to capture light, and the wavelength of light is the color of light used for observation. For example, let's say we have a microscope with an objective lens of Na equals 1.4, and we are using green light of 550 nanometers wavelength. The resolution would be resolution equals 0.61 by 550 Newton meters, 1.4 equals 240 Newton meters. This means that 2 points closer together than 240 nanometers cannot be distinguished as separate entities by this microscope. To achieve higher resolution, microscopes with shorter wavelengths or higher numerical apertures can be used. Electron microscopes use electrons instead of light and have much shorter wavelengths, enabling them to achieve much higher resolution. 3. Numerical aperture, Na. It is a measure of the light-gathering ability of a microscope's lens. It is determined by the refractive index of the medium between the lens and the specimen, and the half angle of the maximum cone of light that can enter the lens. The higher the numerical aperture of a lens, the greater its resolving power and the better the image quality that can be obtained. 4. Oil immersion is a technique used in microscopy to improve the resolution and image quality of specimens. It involves placing a drop of immersion oil with a high refractive index between the objective lens and the specimen. Oil immersion is most commonly used with high-power objective lenses, which typically have the highest numerical apertures, and this technique can benefit because of their increased light-gathering ability provided by the oil. Different types of optical microscope. We can differentiate two types of microscope based on the number of lenses they use. Simple microscopes have only one lens, which is typically a convex lens, a lens that bulges outwards in the middle. While compound microscopes have two or more lenses that work together to produce a clear and magnified image of the object being viewed. Compound microscopes can achieve much higher magnification and resolution than simple microscopes, and are used extensively in scientific research and education. We can say compound microscope is one kind of optical microscope that uses visible light and a series of lenses to magnify and visualize small objects. Stereo microscope, also known as a dissecting microscope. A stereo microscope is used to view larger specimens, such as insects, plants, and rocks. It has 2 eyepieces and 2 objectives that provide a 3-dimensional view of the specimen. Digital microscope. A digital microscope uses a digital camera and a computer to capture and display images of the specimen. Polarizing microscope. A polarizing microscope uses polarized light to examine the properties of crystals, minerals, and other materials. Confocal microscope. A confocal microscope uses a laser and a series of lenses to produce a 3-dimensional image of the specimen. It is used for examining specimens in great detail, such as individual cells or subcellular structures.